Well, everybody, it's a pleasure today to welcome Dr. Tony Firulli for Faculty Fridays Research Seminar Series. Dr. Firulli is the Carleton Buehl-McCulloch Professor of Pediatrics at the Indiana School of Medicine. He's also a faculty member in the departments of Medical and Molecular Genetics, Anatomy and Cell Biology, and Biochemistry. So, Dr. Firulli's and my paths crossed in Dr. Eric Olson's lab about 30 years ago, where he was instrumental in engaging in the discovery of basic Helix-Loop-Helix transcription factors that play a key role in heart development. So Dr. Firulli's group has firmly established that these transcription factors have broad dimerization characteristics and that phospho-regulation of these proteins help define specific bHLH partner dimer choice. More recently, he's done some really interesting work. He's taken the response element that these hand transcription factors drive expression through and used them as a tool to interrogate the role of this enhancer in craniofacial patterning and morphogenesis, which I thought was a pretty brilliant approach. So without further ado, I'll hand it over to Dr. Firulli, and if you could remember to mute your microphones, and that would be appreciated. Take it away, Tony.
Thanks, Jeff. It's really good to be here and see so many people virtually. It's great to see Eric and you. I mean, 30 years ago, it just makes me go, how did that happen? But I am going to share my screen, and when I start my presentation, I don't think I'm going to be able to see you. So for everybody who has any questions, Jeff, I'm going to rely on you to stop me and ask them as they come up, because I think that's probably the best way to go. I will get started right now. So this is just my title slide. And so what we've been really interested in over many decades, and I grabbed this interest in Eric's lab when I was a postdoc, was congenital heart disease, where, basically 8 in 1,000 live births and, 3 to 4 in 100 of stillborn births, what's encountered is a defect in the heart, where this causes problems where in many cases you need immediate surgery to correct. Most congenital defects are well tolerated during fetal life because they're mostly on the right side of the heart where your lungs are not being used. And then when you're born and you need to breathe, this starts to become a problem. The etiology is unknown in many cases, and there are genetic factors, and the treatments for all of this end up being just having your doctor watch your baby to emergency surgery where very talented surgeons basically replumb the heart. Okay, there we go. And so heart development looks pretty cool, and this is just kind of an overview for the people who are not aficionados. If I could start up here, this is a mouse embryo, and this is at E7.5, so this is just as gastrulation is starting to kick in full steam. And this would be the neural tube here, and the heart forms from these plates of mesoderm that are anterior and lateral to this neural tube. There are 2 consecutive fields, the first heart field and the second heart field. And what happens is as development occurs, these fuse at the midline and form a tube where you can see the secondary heart field, the primary heart field, and then the inflow and the outflow that comes in. And through the addition of secondary heart field cells to this growing structure asymmetrically, more from one end than the other, what you get is that the tube loops and then puts or juxtaposes what we know as the right left ventricle and the atria in the correct places. And then at around 10.5 days of mouse gestation, extra cardiac cells like the cardiac neural crest start populating and building the aorta and pulmonary arteries, and septation occurs, and then you have a fully formed organ by 15.5, 14.5, where a mouse gestation is 19 and a half days. And in humans, the time scale is different, but because we're all related in mammals, it's pretty much the same. So for those old enough to remember Donald Rumsfeld, I will use his quote to kind of frame up what we're going to do today. So reports say that something, and in my case, I'll say, let's say congenital heart defects, hasn't happened are always interesting to me, because we know there are known knowns. There are things we know we know. We also know there are known unknowns, and that is to say we know there are some things we do not know, but it is the unknown unknowns, the ones we don't know, And if one looks through history in our country and all of this, it is this latter category that tends to be the difficult one. And so if we extrapolate that to congenital heart defects, there are a number of knowns, and these are generally syndromes, the George syndrome, Holt-Oram syndrome, and these have been associated with genetic mutations and defects within specific genes. And then we have a category of clinical outcomes that we're well aware of, the known unknowns, tetralogy of Fallot, hypoplastic left heart syndrome, transposition of the great arteries. We know they exist, but the mechanisms at this point are anyone's guess. And I end with the unknown unknowns, and it seems the way we move so slowly through time in trying to figure out what's going on is got everything to do with my favorite gene, hand1. So for people not necessarily familiar with bHLH transcription factors, this is a schematic on how bHLH factors work. I've been using it ever since I've been a postdoc. These proteins are, they form dimers and bind DNA collectively, and they are composed of a basic region that actually contacts the DNA, two helices, which are amphipathic in nature, which means that they basically have a hydrophilic side that likes water and a hydrophobic side that does not like water. And the two proteins stick together through those hydrophobic surfaces and create a combined DNA binding domain that binds a very simple sequence called an EBOX, CANNTG. hand1 is a member of the twist family of bHLH proteins, where as a postdoc, most of these were all cloned in Eric's lab, and they share a lot of similarities in amino acid identity. Specifically, they have this very nice region here that is a threonine separated by an amino acid and a serine, which are phospho-regulated by PKA, PKC, and a phosphatase called B56 delta, which changes the charge on this helix here and then allows these factors to form different kinds of dimers, giving them a very broad and resilient way to regulate genes within the same cells and within different cells, depending upon which protein partners are available. So when we look at hand1 expression related to heart development, it's expressed in many other tissues. It is expressed in post-migratory cardiac neural crest, and that's seen here about 9.5. The LV myocardium, this area called the myocardial cuff, which is basically the connecting cardiomyocytes to the smooth muscle of the aorta and the pulmonary artery. And there's a population of cells that are expressed in the septum transversum, which give rise to the epicardium. So the septum transversum would lie just below the heart here. It would separate the thoracic and the abdominal cavity. These cells move into this proepicardial organ, and then at 10.5, well after hand1 is shut off, they form the epicardium of the heart. The systemic knockout of hand1 is embryonic lethal at around E9.5, mostly due to placental insufficiencies. Neural crest and limb conditional knockouts strangely show no really obvious phenotypes. suggesting functional redundancy or a lack of importance in those tissues. So I'm going to start with this picture here because I think it kind of typifies how we kind of gotten interested in this. And so this is a cardiomyocyte left ventricular specific deletion series using hand 1 conditional and hand 2 conditional mice. And what we see is that there are genetic interactions between both hand 1 and hand 2. And that could be because they're doing the same thing in cells. It could be literally that they're forming a dimer together and doing the same job. It could be they're competing for another bHLH protein and that difference is taking place. But if you look through from left to right, left where you have the most hand gene copy number and right where you have the least or 0, what I hope you could appreciate by these beta-galactosidase pictures showing the lineage from a Rosa beta-gal allele is that you can see beautifully the left ventricular expression in a control mouse. And if you look below at the sections, you can see all the trabecular blue, the compact zone here is blue. And as you start slowly taking hand gene dosage away, especially here in a hand 1 knockout with just 1 copy of hand 2, and certainly in the double, what you can see is that the left ventricular structures are there, but they don't look right. You can see that where there should be mostly open space as a lumen, that they're kind of compacted with myocardium, and that's a not normal thing to do. And in fact, in the outer layer, you can see that this is brighter blue than this, not because there's less beta-galactosidase staining, but because the staining has moved from the outside of the heart to the inside of the heart. And so looking at this data, what are the genes within the regulatory network that are modulated by hand factors was what we were after. So what we did is we took a fairly common approach.
When do those mice die?
Do what?
How long do those mice survive?
The doubles. The doubles actually live to birth. And they have conduction issues, and we can rescue them by crossing those double mutants onto a diphtheria toxin mouse, because the proliferation occurs, as you know from your work, all the way up until 3 or 4 days after birth. And so basically all the cardiomyocytes in the left ventricle that don't express hand 1 or hand 2 or the mutant, those cells all die, and then the remaining cardiomyocytes just rebuild a normal ventricle. We put that out in PLOS Genetics in 2017, I think. So to kind of go through this, we generated hand 1 knockouts using a key transcription factor and cardiogenesis, NKX 2.5, where this is a knock-in Cre, which is important because one allele is going to be a Cre and one is going to be the NKX 2.5, and there are caveats to that. We isolated poly-ARNA from 11.5-day ventricles, and we simply did an RNA-seq analysis followed by bioinformatics. And what we found in this, we found very surprising, was that during heart development, the gene programs or the IPA categories that had the highest Z-scores with the most genes that had the highest functional or false discovery rate for change were all in programs about adult heart failure. So NFAT and cardiac hypertrophy signaling, TGF-beta signaling, renin-angiotensin signaling, protein kinase A signaling, which is interesting because the post-translational tie-ins, and they're all pretty robust Z-scores here. Interestingly, the one that is up, and it's certainly up a lot, is EIF2 signaling, which is involved in protein translation, which may fit into what I'm going to show you in the next couple of slides. So we did a little bit of validation of this just using QRTPCR, and I always like to show people home mounts. So this is a NKX 2.5 hetero, hand 1 hetero, showing that, hand 1 is expressed in the left ventricle by RNA, and that when the Cre is on a homozygate flox-flox background, the expression within the lower jaw, or the first branchial arch here, is still maintained, but the left ventricular expression goes away. And then this is just a different Cre called alpha-myosin heavy chain Cre, and the result is the same. We see the hand 1 expression reaches a very low level, and that's shown here in QRT-PCR, where we can see hand 1 levels plummet in this mouse model. Hand 2 levels are not changed. But many of the genes that are involved in the pathways we saw, cited 1, ANF or NPPA, were affected negatively. A few interesting proteins were upregulated, like TNNI2. And then what we also noticed was there were some genes in the cardiac conduction system, a potassium channel HCN4, and a gap junction protein connexin 40, which were also significantly down, suggesting potentially maybe we had some tie-in to cardiac conduction. Hand 1 and hand 2 play an important role in sympathetic neurogenesis. And so we were thinking, well, maybe this is some kind of specialization cue for subtypes of cardiomyocytes as we move forward from here.
Hey, Tony, it's Piano.
Yeah.
Can I interrupt you for a sec? How you doing?
Yeah.
Hey, I think you alluded to it just a couple slides ago, but is there a haploinsufficiency with that Cre driver?
Yes.
The NKX25, and it doesn't look like it. I mean, I'm not an embryologist, but it looked like those controls look normal. But
it's all about penetrance, and they're subtle. I'll get into that in a few slides. So
how many of those targets? How many of those genes are direct targets?
Well, so we're hoping to find that out directly within the next few months with the new mouse that we have. But I think that is the biggest question, because without DNA occupancy data or really good enhancers that you can test in mice to try to see whether or not they can be activated or they get affected directly in like a hand 1 loss of function model. That is definitely our goal, to try to push forward with that. So we then took these mice, and I wish I could say we let them go to birth, but in reality what happens is you miss plugs and you miss litters, and then you go and you look at your experimental cages and you see pups, and so you genotype the pups and you say, oh, we'll just use whatever we get for the next round. And what we found was that we got a fairly normal amount of Cre positive hand 1 flox-flox mice. And so these mice are on a mixed background, and so we were like, okay, let's see what these hearts look like. And so we took a couple of the adults and opened them up, and we were shocked to see that compared to a normal looking heart, where you could see the right and left atria, the right and left ventricle, a nice kind of apex, that many of these hearts were huge. And the left atria was very hypertrophic as well. And so we were like, what are we going to do now? So we looked at these a little bit closer by section. And I believe these are the same hearts that I just showed you. So you can see this very nice, normal looking wispy right ventricle, the interventricular septum, which goes all the way up into this membranous cap, keeping the right and the left side separated, and a normal muscular left ventricle with the atria here. What we see here in the mutant is a highly muscular left atria, hypertrophic muscle everywhere. We see that there is a ventricular septal defect in this surviving mouse. And an interesting thing that I will play on a little bit more, this is a papillary muscle. So papillary muscles are what articulate the ventricular valves. And this particular papillary muscle is driving directly in the mitral valve, or the valve of the LV, which plays into with the phenotype ends up telling us. These are lungs, and this is a normal open lung. This is a lung full of fluid, and these mice are identifiable in the cage because they just sit in the corner and pant because they're undergoing heart failure, and this is a full-blown cardiomyopathy. So because we had access to echocardiography, we took some looks at these mice that were alive. This is a schematic of an outflow tract, and you can see here in the still image, the pulmonary artery, the right ventricle, the tricuspid valve, the right atria, and the aorta are all marked, and this is kind of what it looks like, and the blue obviously is lung flow, and then the red coming back is from the lungs. We can see here in this still image by the yellow caret or the yellow arrowhead that there is a break in the aortic wall here, showing that there would be a VSD there. This is a deeper optical or a deeper sound section where you can see the right and left ventricle next to each other, and this gap in the septum is a muscular VSD. We can look at systolic and diastolic function, and this is a measure of an older mouse that has an ejection fraction of 39% and a fractional shortening of only 19% after a year. It is very sick. And then if you measure flow over the mitral valve, and I'll spend a little time on this. So this is called EA reversal. The first wave that you see in these little snippets that look like M's are, this is the E wave, the first one, and the one to the right is the A wave. The E wave represents when the mitral valve opens and all of the blood from the left atria just falls into the left ventricle by gravity. The A wave is when the atria gives a little twist and a squeeze to kind of wring out the last bit before the ventricles start to contract and push the blood out through the aorta. So in a normal condition, the E wave is always taller than the A wave because the gravity gets most of the blood out, but here they're balanced. So our echo machine has color Doppler, and so we wanted to be able to see whether we could find these VSDs in a living mouse and actually demonstrate one. So just like your weatherman showing you the thunderstorms coming in or the tornadoes coming in, the color Doppler from the wand. So what you're looking at in this still image here is through the chest of an anesthetized mouse. And so flow coming to the probe is in red, and flow moving away from the probe is in blue. And so what you can see here, where these arrows denote, is that there is a nice little hole here where the blood is pushing out of the aorta at the top of the heart. And when we measure this using pulse wave Doppler, which is a slightly different technology in the ultrasound machine, we can see that there's a high velocity jet that is at 1,800 millimeters per second. And so obviously this is not lethal, but it's certainly not a normal condition. So getting back to where I showed you in the section that the papillary muscle was weirdly attached to the mitral valve, we ran across a clinical condition that's called mitral arcade. And this condition is very rare, and it's represented by either short or missing chordae tendineae, which are these fibrous puppet strings that when the papillary muscles contract and relax, articulate the valves into open and closed position. So what we thought we were seeing then is potentially the hand-1 knockout model being a model for this. And so we looked using a stain that picks up fibrotic tissue. This is serious red, fast green, where the muscle is in green, and the fibrotic tissue, like the valves, are seen in red. And you can see here, looking at this magnification of this whole heart image here, that we have papillary muscles here, and you can see the muscle driving right into the valve where there's no gap or space in between. And you can see here, and actually this echo still image, here's a mitral valve, here's the left ventricle, and we have a papillary muscle that is like contiguous with that valve. We can see here that the muscle has a lot of fibrosis around the outside, which is an indication of failure. And just because it was in this mouse, you could see here that there's a nice muscular VSD that is in the lower base around this version here. So these mice have a lot of interesting problems. So to get back to flow efficiency of the NKX allele, this is a really known thing, and that heterozygote NKX 2.5 mice have a subtle but large complement of congenital defects that we wanted to be sure was not a problem, and was fooling us into thinking hand one was doing more than it was doing. Especially since, this is going back to the data I generated in Eric's lab when we had first made the hand one knockout mice, where because I inserted a beta-galactosidase gene in the null allele, we could track hand one expression. And you can see here the left ventricle, the outflow tract, and then this lateral mesoderm here. Well, we crossed it onto the NKX 2.5 systemic knockout, and what you see is that there's no more cardiac staining of hand one. And so that could indicate that NKX is in some way influencing hand one expression. It could simply mean that these left ventricular cells no longer exist, and so they're not there to express hand one. But either way, this genetic interaction could potentially be confounding the way we look at things. And so we had a way around this, and we used the alpha myosin heavy chain Cre. And I'm just re-showing you here on how this deletion works. And we looked at mice that were homozygote hand one knockouts using that Cre, which does not have a genetic effect. And you can see here a nice section through an adult heart. These were also found at Mendelian ratios. The atria look great. There's a patent septum, RV and LV. But when we look at these mutants that we see, again, a very large left atria, we see a lot of tissue in the lumen of what should be the left ventricle, papillary muscles going to the wrong place, and it does not look good. And in a different mouse that we fast green and Sirius red stain, here's the inventricular septum that has a lot of fibrosis. You can see here very, very clearly that the papillary muscle is just driving right into this mitral valve. So the phenotype seems to be independent of whether NKX 2.5 is haploinsufficient. So we then knew that these mice lived to about a year, year and a half before they died. They died earlier than wild type or heterozygote littermates. So we wanted to see where and when we could start seeing the problems with cardiac function. And so we started with a 4, 8, and 12-week analysis of knockout mice. We used the NKX Cre by itself as a control, as well as just wild type control. So the wild type control is in the light gray, the NKX 2.5 is in the open, and the NKX hand 1 is in the darker gray, because I didn't want to pay for color images. And what you can see here in these scatter plots is that systolic function, when the heart is squeezing, we did not see any measurable difference in fractional shortening or ejection fraction in these mice. When we looked at diastolic function, when the heart is relaxing and you get that open drain into the left ventricle, what we saw was aortic peak velocity was altered. Pulmonary peak velocity was not. And so these mice have an issue, a noticeable issue in aortic peak velocity. And so that gets us back into the EA wave story. And so we did some calculations, and I'll just say here that, clinically the E to E prime ratio is related to atrial pressure. It can show that increased filling pressure in humans gives diastolic dysfunction, and this is assumed with a clinical number in humans of a ratio that exceeds 15. What we see here by all the statistically significant highlighted tables is that these mice have issues with mitral valve function, and they have stiffer myocardium around the area where the mitral valve sits. So they have diastolic dysfunction. And so I stole this from a medical text. It's pretty good, and I was really happy to read it when I found it. What is diastolic heart failure? Well, there are different grades of it, and in grade 1, we can see that mitral inflow Doppler echocardiograms will show that there's a reversal of normal E to A ratio, and I'm like, hey, we see that, and this pattern may develop. It could change in mice from being normal to back and forth, and grade 1 patients may not have any clinical signs or symptoms of heart failure. And grade 2 diastolic dysfunction, also called pseudo-normal, this is associated with elevated left atrial filling, and this is associated with left atrial enlargement. And I said, hey, we see that too. Isn't that interesting? And when you go down to class 3 and 4, just to try to keep on time, I'll just go to the punchline. These patients have left atrial enlargement and reduced left ventricle ejection fraction. And that's what we see in these older mice, that they start with normal systolic function, they have issue with mitral valve mechanisms, and that when they get older, their hearts just get progressively worse and they go into systolic heart failure, just like in people, which we thought was really cool. So we then started asking about cardiac conduction, because the force, of how the heart contracts can have to do with the muscle strength and the layout of it, but it could also have to do with the wiring that's triggering the heart to fire and contract. And so we wanted to see whether or not there were issues with this system, because we had genes in our RNA-seq data that were telling us there might be some problems. And so we did 2 simple things. Well, one simple thing and one more complicated thing. We did electrocardiograms on anesthetized mice, and then we took hearts out and hung them in a Langendorf preparation, loaded them with a voltage-sensitive dye, and did optical mapping. Whereas the conduction system fires through an electrical probe, you can see the flash from blue to red, showing where the flow of voltage goes through the heart. So we did this, and I apologize, this is a very big and busy slide. We'll focus on the ECGs first, and the 3 different leads that are measured. What we found in all 3 of these leads was that the QRS complex, the ventricular contraction complex, was extended. It was longer, so it's called a prolonged QRS. What we observed in the optical mapping was that we had a condition called right bundle branch block, or RBBB. And the phenotypes were variable depending on genotype, but it was very consistent that the cardiac conduction that we measured in these mice was abnormal. So to summarize this part of the talk, we wanted to dive into hand-1 gene regulatory networks. And so through the use of RNA-seq and an IPA analysis, we found a number of pathways that were affected in these mice early on before there's any indication of such problems. Cardiac hypertrophy and cardiac conduction issues and failure categories. The hand-1 cardiomyocyte loss of function is survivable on the genetic background that we happen to have. And the phenotype includes hypertrophy of the muscle, ventricular septal defects, and a papillary muscle abnormality that resembles a very rare clinical condition called mitral arcade. Collectively, these defects cause diastolic insufficiencies, which result in heart failure with preserved ejection fraction initially. And then we also observe conduction system gene expression defects, as well as actual conduction defects. So at this point, when we were looking at all of this, we had to recognize that the conduction phenotypes that I just showed you could actually reflect a role in the conduction system, the CCS, the actual wiring that's doing this. But it could also very easily be secondary to the consequences of the altered muscle morphology. So if the heart is formed so badly that the conduction system doesn't articulate correctly or does not actually hit some, key places where cardiomyocytes need to talk, you can have conduction defects just because the structure is wrong. And so we then wanted to figure out whether it was one or both. And so we started learning about this. This is the schematic of how the cardiac conduction system is set up in a heart, where you have the sinoatrial or SA node, the AV node, and then right here is the His bundle. You have the right and left bundle branches, which go down the interventricular septum, then break off into Purkinje fiber networks. And when you talk about an EKG, you can see that the P wave, the QRS complex, and then the T wave. It is this QRS complex that is obviously looking like it is not normal in the hand 1 loss of function mice, and this represents the ventricular contraction. So one of the things we stumbled across that got us even more excited was that this is a paper that was published in Nature Genetics, and then this is just a table of this data summarizing all of these different genes that were associated with conduction defects through genome-wide association studies, that lo and behold, QRS duration had some hits in hand 1. And so the QRS interval is prolonged when it's longer than 120 milliseconds in patients. And so this is now something that we are like, wow, this is really great. But this single nucleotide polymorphism, or these 2 SNPs that were found in hand 1, were well upstream of the exon. And so it wasn't like a point mutation in the hand 1 protein. We then suspected or hypothesized that this was in potentially a transcriptional enhancer that could be specific for the conduction system, or certainly one of the enhancers that we had identified in the heart. And so when we looked at this, we found that it was very close to our hand 1 LV enhancer. And this is data basically showing that we have this area that's about minus 15 kb upstream of the hand 1 transcriptional start site that we can whittle down to about 300 bases and get left ventricular specific expression. This is the hand 1 knockout that we made, that Eric and I made many years ago, and we can see that there's left ventricular expression. We can also see that there are neural crests that are populating the outflow tract, and the aortic septum is also full of neural crest cells. This enhancer only expresses within the left ventricle. And we found 2 very evolutionarily conserved GATA cis elements that when you mutate these sites and you make transgenic animals with the point mutants, you get no expression within the LV. So this enhancer is totally dependent upon the activities of GATA transcription factors. And these are the 2 GATA cis elements where we're just using an old-fashioned gel shift or an electrophoretic mobility assay, where we use programmed rabbit reticulocyte lysates that produce a protein, and in this case, GATA4. When we add the probe, which is very small, that's radiolabeled, it runs through a polyacrylamide gel very fast. But when some of the probe binds to the protein, it slows the migration of the probe down. So you can see this very nice band here. When you add a ton of cold probe that hides the radiation, this band goes away. But when you use a mutant sequence, which doesn't bind the protein, it maintains itself. And so that's how that works. And so I'll make a disclosure. I am friends with Dr. Miano, and for the last 3 years, Dr. Miano has been speaking to me about CRISPR. So we had this enhancer, and we decided we wanted to know what it was really doing. And so we took Joe's advice and generated a CRISPR strategy to just delete this from the mouse genome. And this is a whole mount in situ hybridization showing this is the outflow tract here. This broad expression here is the first pharyngeal arch, which is the lower jaw of the mouse. This is the lateral mesoderm in the umbilical area. Here is the LV. And what we can see in mice that are homozygous for our delta LV allele, all of this expression is completely fine, and it's the left ventricle expression that goes away. And then this is just some QRTPCR showing that, in fact, we see that hand 1 goes way down, GATA4, which is upstream, is not affected. But 2 of the genes that are downstream in our RNA-seq data set and that are involved in cardiac conduction are down, connexin 40 for sure. So we got excited about this. So we looked at these homozygous delta LV mice, and they lived to adulthood. And this is a nice cross-section of 2 hearts, one from a control and one from a delta LV animal. And what you can see here is that, you've got nice RV chamber, LV chamber. You can see a nice papillary muscle and a chordate tendidae here driving up to that mitral valve. The septums are patent. The atria are normal sized. And so this mouse does not have any morphological defects that would drive you to think that there were problems in cardiomyocyte-mediated heart development. So we went back to doing ECGs and optical mapping. And here are the ECG data. And what we found here is that these mice exhibited a shorter PR interval, which was new for us, but they also had an extended QRS complex. And we got kind of smart and started putting cartoons in our figures. This is atrial pacing, where you put your electrical probe at the top of the heart by the atria, and then the electrical signal fires down the sides of the septum, and then you have 2 epicardial breakthrough points where the signal then rushes up through the myocardium. And then by pacing from the muscle itself, that measures not how the current moves through the conduction system, and measures how it moves through the muscle itself. So here you can see in this control heart where you see these 2 epicardial points. The time scale here is 0 is very fast and red is slow. And so we see here that we have this condition where you have the right ventricle and left ventricle giving you the 2 breakthroughs you expect. And the delta LV mice, you see an LV breakthrough, but no RV breakthrough. So the RV breakthrough to the LV is delayed or it's completely blocked, similar to what we saw in the cardiomyocyte-specific deletion. So just by taking out the left ventricular enhancer, we are seeing very strong conduction defects. So going back to looking at things, which is what developmental biologists love to do, we took advantage of our hand 1 lag Z allele again and just cut hearts open and stain them for beta-galactosidase. And what we saw, which was quite an exciting surprise, was that the beta-galactosidase staining was staining what we looked like conduction tissue, Purkinje fibers. When we put this null allele on the LV deletion allele, what we saw was that there was a lot more blue staining, that it looked hypertrophic, and that was not normal. We then took delta LV mice and heterozygous LAG-Z mice and did immunostaining for a gene called contactin-2, which is expressed exclusively within the cardiac conduction system and comes on at about 2 days after birth. So these are adult hearts. What you can see here is that the contactin-2 is marking at the top of the interventricular septum what is the His bundle. So the right and left bundle branch kind of form an upside down Y, where the His bundle is the stem of the Y. And in our delta LV mouse, you can see that not only is the contactin 2 staining more broad, but rather than having this nice tight package of cells here that are hand 1 positive, you see that these hand 1 positive cells still exist, but they are not organized very well. And you can see here the Purkinje fibers going down the side of the left ventricle. This is the left bundle branch. So there's some morphology things going on. We wanted to be more careful because we were worried that by taking away a hand 1 allele on the delta LV allele, what we were seeing was transcriptional feedback, where because there wasn't any hand 1 in the LV, the transcription machinery on the beta-gal allele would try to drive more hand 1 to compensate. So everything was more because of this real effect you see when you study transcription. So we switched to looking, and we have the contactin 2 IRES Cre. So this mouse has no haploinsufficient issues and expresses Cre very nicely. And what you could see here, the controls of the right and left side are on the left side, and here the delta LV mice are here, where you have the left and the right ventricle opened up and you're looking into the luminal chamber. You can see quite clearly here that there is a propensity of hypertrophy in the conduction tissue marked by this Cre and the delta LV mutants. We went a step further with the help of a good colleague and friend, Vincent Christoffels in the Netherlands. What we did here is we did in situs on an entire heart for another gene called HCN4, which is also specific for the cardiac conduction system in the adult. We then took every single one of those in situs and then 3D reconstructed them using a computer algorithm where you could actually generate a 3-dimensional image of what the conduction system looks like relative to any heart that it's in. These are the images of these two conduction system maps in the hearts. And then what's a little clearer is when you take the cardiomyocyte structures away. Here you have the His bundle and the right and left bundle branches, and then this wing that goes back underneath the left atria. They're all running straight down the septum. Here we see that this is very hypertrophic. We see that they're running more parallel with the septum versus running straight down, and that the tissue is definitely not organized correctly. So here we are, and we were all excited about the fact that these 2 mouse models now have conduction defects. But when we go back to the human data that showed us that there were instances where hand 1 was mutated and associated with these kind of problems, we were really sad to see initially that here are the human SNPs, and here is the location of this evolutionarily conserved enhancer. And so these 2 SNPs are not conserved in mouse sequence. So we were left with a puzzle on how in the world these were picked up as being sensitive to problems here in a human being when they're not here in a mouse. And so what we did was we just called the experts who did the GWAS study and asked them if they could do us a favor. If they could just look very carefully between 478 and 595 and see if they could just find anything else that was in here that would help us. And so they did that for us happily. And what they found was that there were 3 additional SNPs between these 2 bookends, which are marked here. And this is the conserved region that's underlined in the super conserved region in gray. So there were 3 more SNPs that were found in between those that lie inside the enhancer. These 2 right here are very highly conserved sequences within the enhancer. And this one in particular was in one of the GATA sites. Here's the 3' GATA site. The GATA sites are in green, and this does not say GATA because it's backwards. It is GATA on the opposite strand. This one right here that you see has that SNP right here that has a linkage score that's completely ridiculous, that if you get these two, you always get these three. So it's a family of 5 in people. So that was like, wow, this is cool. So, oops, sorry. We also then decided to use CRISPR again. And what happens if we just make a mouse that has these conserved SNPs in it? And so we set off to do that. So we actually made a mouse, and here's the sequence right here, where we put the 36 and the 375 into the mouse genome. We also altered this base right here because it formed what's called a PAM, and that would cause problems. But we used a conservative. This in the evolutionary conservation pileup is either a G or an A, and so we kept it very conservative. And we got this mouse. This mouse is kind of interesting. This is wild type hand-1 expression in the red bar or the maroon bar. And the green bar represents the expression level of hand-1 that we can detect in the delta LV mouse. So it's well below 80% of normal hand-1 expression, but it's still there a little bit. We only see a 20% drop in hand-1 expression and the 3675, 3675 SNP mice. And we were very surprised by this because it was really for us, wow, this is not even close to being haploinsufficient. It's not even as bad as being a heterozygous. But then we looked at the data and we decided to do some analysis. We knew that these sequences were binding GATA4 and probably other GATA factors. And so we did a gel shift using the SNPs. And what we found here was that in the wild type condition, we could get robust GATA4 binding. We could compete it off with wild type cold oligo. It would not compete off with mutant oligo, and it would not compete off very effectively with our SNP oligo. We switched and then labeled our SNP oligo, where this one only has the 75, which is the GATA site, and then this one has both. And what we see here is that we do bind GATA4, but it binds far less efficiently than the wild type sequence when this is normal. Looking at ECGs from these mice, we didn't see anything that was really jumping out at us. But when we went to optical mapping, we saw some interesting things. So we see that here are 2 epicardial breakthrough points in the RV and LV. We see some strange things in the hand-1 3675s. We see here a single breakthrough point in the RV only. Here we see 3 breakthrough points, 2 in the LV, 1 in the RV. And then when we do this by muscle, where we put the probe on the apex of the heart versus at the top at the ventricles, or at the atria, I'm sorry, that there's no phenotype observable in the current running through the muscle itself. It's getting the current to the apex at the right time and at the right place. And then here are some more mice here where we see 2 in the LV. You see this kind of delayed apex and the breakthrough in the RV here, and then there are 2, and we get a collection of mice that look normal. Some have single RV, some have a double LV, and 1 RV, some have only the 2 LV, and then we get a few mice that have normal. So there's a spectrum, but nevertheless, they're exhibiting phenotype, which we were really excited about. And so we have a GATA-dependent hand-1 enhancer that seems to be necessary and sufficient to drive the LV-specific embryonic expression of hand-1. That in humans, there are human small single nucleotide polymorphisms that are associated with QRS defects. When we delete this enhancer, we get prolonged QRS duration, decreased ventricular conduction velocity, and in most cases, delayed RV breakthrough. So this is a right bundle branch block, similar to what we saw in the straight knockout of hand-1 protein using a cardiomyocyte-specific CRE. The SNPs that we found 375 reduces GATA4 DNA binding in EMSs, and that these mice present with abnormal epicardial breakthrough patterns, supporting the necessity of normal hand-1 transcriptional activity for conduction system morphogenesis and function. And our follow-up studies are really trying to kind of figure out how to get a good understanding of how these downstream targets that hand-1 must be articulating within the CCS are working. And some of that we can see, I can show you what we're planning to do is we have been looking at a number of genes. This is connexin 40 here. This is the potassium channel HCN4, and these are both genes that were regulated in our RNA-seq. And we see differences in the delta LV mice. So these yellow boxes represent conserved non-coding regions that you can assay using a website or a set of tools called eMERGE, where we can find hand2 and NKX 2.5 DNA occupancy peaks or ChIP-seq peaks. We don't have hand1 ChIP-seq yet, but it's coming. So then you can see that this region here has broad overlapping peak. When this is brought into a condition where you use a heterologous promoter to drive a HSP 68 to drive a beta-gal, and you make mice with these, both this connexin and HCN4 enhancer that are dictated here are expressing in tissue where you would likely observe hand-1 regulation in the LV. And there are actually human SNPs associated with conduction defects in some of these tissues. And so we will slowly try to build together this gene regulatory network within the conduction system where hand1 is turning on factor A, B, and C, and how those things collectively manifest into the phenotypes we're seeing. So the last thing I'll go over, and this is pretty new data that we're very excited about, is answering the question of hand1, where are you? So looking at mRNA is always great, but to know where you can look and find the protein really tells you how and what it's doing. And so this mouse was very difficult for us to make because we had a number of unforeseen problems with the way it went together. But what we did was we put an amino terminal flag, triple flag tag on the amino terminus of hand1, and we knock that in. And so these mice then can be immunodetected with a robust antibody versus the not so robust antibodies that we have for hand1. Keep in mind, transcription factors are usually expressed at very, very low levels compared to, say, like a cellular structural protein like an actin or a myosin. So it's always been a challenge to break through the background to signal noise to really believe that you're looking at something that is real. So we made this mouse. It's viable as a homozygote, and you can see here, this is the aortic sac, where you can see the neural crest cells that are populating the outflow tract of a 10.5-day embryo, and this is kind of a lower view in a different section. You can see the pericardium where hand1 is expressed here, the mix of neural crest cells that are moving in here. This is a beautiful left ventricle. It's a little ripped, but you can see here beautiful nuclear left ventricular staining, far more than what you see in the right ventricle. And this is the lower jaw, the first pharyngeal arch, where you can see the hand1 protein in this distal cap where it's supposed to be expressed, around the umbilical artery where it's supposed to be expressed, and then some areas in the hindgut and the vitelline artery here. So we now have this tool where we're going to do two things. We're going to actually find out where hand1 is occupied in the genome. And then we can then take where, and this is directly answering Eric's first question, where now we can look at genes that are regulated, see whether or not hand1 occupancy peaks are in that local neighborhood. Look and see where the contacts are made, if these are evolutionarily conserved regions, and then pop them out and test them for enhancer activity, validating that they are in fact part of this gene regulatory network. And then we can subsequently delete those enhancers by CRISPR and see how many of the phenotypes that are observed by knocking that gene out just where hand one is regulating it, how many of those phenotypes correspond to what we see from hand one. The second thing we're going to do is we're going to see who is hanging out with hand one. And we're doing an experiment right now called an IP mass spec, where we're going to take various tissues where hand one is expressed, most certainly the heart. We're going to IP the hand one protein with flag, and then we're going to mass spec everything that sticks to it. We expect to see E proteins. We expect to see, hand one. We expect to see potentially some kinases and some phosphatases based on what we know on how hand one is functionally regulated as a protein. And we hope to get some surprises that will potentially help us fill in the gaps of why, when you knock this gene out, there aren't super severe phenotypes, and its role in transcription doesn't seem to be specifying tissues to differentiate definitively into something. Maybe what it's doing is controlling mesenchymal nature, or whether the cell is a polar cell or not a polar cell, or whether the cell is, going to be able to move or not move. These are kind of the questions that we're very excited about answering in the next few years with these tools. So I will end by giving credit where credit is due. The bulk of this work, the first part of my talk, was done by Beth, and the conduction and the sit work was done by Josh Vincenz, who's just taken a job at Regeneron. Then Danny, who is our lab manager, Rajani is a postdoc, and Gabe is a graduate student. Michael Rubar is a colleague of mine here at the Well Center, and he is my expert cardiologist, EP guy, electrophysiologist, and Jury is the guy who works for him and helped. Then there's Vincent in the Netherlands, who did those beautiful 3D reconstructions with the help of Corey. And then Nona and Dan are the GWAS people who got us thinking about this seriously in the first place by associating hand one with conduction defects. And these are all the people who have given me financial support. I'll thank Eric, Joe, and Jeff, because they've certainly given me a lot of collegial career and human support over the 30 years that we've known each other. They've been great colleagues and better friends. And it's been a privilege to be able to accomplish this, because it wouldn't have happened without a lot of other people pushing me up along the way. And I'll stop there and thank everybody, and look forward to some questions.
Thank you so much. That was amazing. Quick comment would be the intestinal fortitude it took to make that triple flag tag hand knock in mice is nice. I really appreciate it. I bet your results are company dependent. Rabbit monoclonal, triple flag tag antibodies. Eric, I see you have. Go ahead.
Hey, Tony, I got a couple questions. Great talk. I love the human SNP data too, by the way. One of my questions, it relates to a paper that was published by Ning Liu a long time ago, in which she revealed that there were DNA binding dependent and independent functions of hand transcription factors. I wonder if you have any thoughts on the extent to which non-DNA binding activities could contribute to the phenotype, and even whether hand factors are always activators or whether they're also repressors.
So those are very good questions. And my feeling is that, in fact, both hand one and hand two, and likely twist and paraxis, sclera-axis, the whole family, are going to have some DNA binding independent functions, simply because they can partner with other proteins, and they can keep them out of play. So, when I used to teach a transcription class in my younger days, And if you just look at 3 bHLH proteins within the same cell, all of them want to form dimers because they don't want that hydrophilic surface in contact with anything that is water related. And so they're going to dimerize with whatever they can. They're going to stick. And so let's say HAN2E12 is the dimer driving a very specific protein, a program in a cell. If there is a change in the level of HAN1 expression or a post-translational modification in HAN1 that makes it more favorable to bind with E12, it's going to steal E12 away from HAN2. And the effect, even though you think it's a gain or loss of function of HAN1, actually has to do with the other bHLHs that have nowhere to go. We published a paper a while ago that when we make different dimers, they have preferences for different E-boxes. So you can shift programs very subtly. And so by taking a factor out of play, and if nuclear localization is a regulatory mechanism, if you have a protein that makes a dimer and then leaves the nucleus, you're clearly getting rid of it. And so I think that is a very likely mechanism. On top of that, there are
my other question, just real quick, is your LV enhancer deletion. It's very interesting, but surprising that it doesn't have a major effect on left ventricular growth. So have you identified a redundant enhancer that's picking up that function?
So we have not. There is a potential enhancer that could be expressed in the LV, but there's two pieces of evidence that we have that I think mean that there's not one. The first one is that when you look at the Crispr deletion, we're really knocking down HAN1 expression by 20%. The second, I think, more convincing piece of data we published in 2017, where we made a Cree with this enhancer, and we crossed it to the diphtheria toxin A chain Rosa mouse. So because of the fact that HAN1 comes on in the embryo and shuts off for the most part in the embryo, as you know, you have this window of cell death that exists between, in my mind, E8, 8.5 to E13. We did that experiment, and at 11.5 and 12.5, the left ventricle was extremely hypoplastic, almost non-existent. But by 16.5, you could not tell that the left ventricle was affected at all. So the remaining cardiomyocytes just regrew. The data from our LAC-Z mouse, from the Cree mice, both the Cree knock-in to the whole locus and the LV Cree, clearly show that not all left ventricular cardiomyocytes express HAN1. It's a subset. And from your data, it's clear that these cardiomyocytes, when influenced by injury, can respond up until late in the neonatal period. And then they become recalcitrant to regeneration. So because that window of time exists, what I feel is happening, that even with HAN1 being all messed up, if those cells could be killed, the other ones just regrow, the ones that don't express HAN1. And in fact, the double knockout, where we knock out HAN1 and HAN2 in the LV collectively, that's a much worse phenotype than just killing all of the cardiomyocytes with the HAN1 enhancer. And in fact, we can kill all the cells with the HAN1 enhancer, and that rescues the double knockout, because the remaining cells that don't have the double knockout just remake a new ventricle. So
Tony, can I just follow up on Eric's question real quick? I didn't have, go ahead, finish your thought. You have a thought to finish.
No. I'm happy to hear the question.
For the hyperplasia, that was a question I was going to ask about. And so do the hand factors intersect with YAP-TAZ, or is it completely independent?
Can you say that again? There was like this big feedback.
The hyperplasia, right, that you were just talking about, is that independent of YAP-TAZ? Do the hand factors intersect with YAP-TAZ in any way?
So, it's funny. There are evolutionarily conserved TEAD sites in the hand 1 LV enhancer. So we initially thought this was very likely. We've mutated them, we've deleted them, and it doesn't seem to affect the expression. That's not to say that they could not have a significant role in modulating and maintaining hand 1 expression if hippo pathway is allowed to put YAP and TAZ in the nucleus so that it can conjunct with TEADS. So, and I look every once in a while for TEAD chip-seeks to see if hand 1 comes up as a peak. They may help drive the right levels of hand 1, potentially hand 2, but they certainly are not necessary, because when we delete them, we get normal, or not necessarily normal, but we see consistent LV expression using those as reporter drivers.
I guess where I was going, and this is naive because I'm not in this field, but I was thinking that perhaps the hand factors were somehow repressing YAP-TAZ, because you said that normally, right, there's massive apoptosis, there's fascia in the ventricle, so you don't get that in hyperplasia.
So what's hard for us to really put a finger on when we're looking at the expansion of the cardiac conduction tissue, as well as just the hypertrophy, whether or not it is just the cells are bigger, or whether there are more cells, or both, because they're not mutually exclusive mechanisms. And, that's not our forte to look at that kind of stuff. And, when we've tried to do the outlines of the cells, we've never gotten like a data set that we've been really happy with. We should go back to doing that to see that. But there may be more conduction cells. It could just be that the conduction cells that are there are more robust, and therefore you see more beta-gal in them because they're driving more gene expression. But the one thing I didn't tie into my talk is that regardless of whether it's more cells or more hypertrophy, going back to that original RNA-seq data set where we see the EIF2 pathway going through the roof, what you do need to get bigger or more is more translation. And what was clear from that data, and we don't 100% understand it, like lots of ribosomal genes, lots of other EIF kind of components are upregulated in those mice in some kind of compensation or messed up direction that they're taking. And so we're trying to follow up on that. But me, I'm just a tiny lab, and so we can only do things in baby steps. But it's exciting to know that I've got a lot of interesting things to do for the next 5 or 6 years.
I see Dr. Wolf has his hand up. Dr. Wolf, go ahead.
I think you're muted, sir. There you go.
Yeah, sorry about that. So I am out of my element in terms of embryology, things like that. But among the things that I teach medical students is that angiotensin II inhibitors are lethal during the latter stages of pregnancy. And we also teach that that your hypertrophy is due to a consequence of like high blood pressure. So I'm wondering to what extent, for example, in your embryos that you have crosstalk between the kidneys, that you might have hypertension in the embryo that's causing the hypertrophy, that you might have altered levels of potassium that are contributing to your aberrant EKG signaling, although I agree that you've got disrupted conduction signals or pathways.
Yeah, so that's a good question. And interestingly, our RNA-seq IPA analysis did pull out renin-angiotensin signaling as a program that was altered in the embryos. It's clearly going to be an effect. ANF or NPPA is clearly a salt modulating factor, and it plays a role in doing volume control and pressure control, if you will. And so there is clearly some ties to that program. Hand 2 has some very nice ties to shear stress, which is also kind of a reactive response to wall growth and trabeculation through its role in the endocardium that we're pursuing, and that I didn't have time to talk to you about today. But there is clearly a possibility that all of these things are affected a little bit, and synergistically you you have this kind of snowball of little tiny 1,000 cuts that, you know, really causes you to have some issues. And, you know, if you think about development per se and evolution per se, you know, big, horrible, on, off, yes, no type of defects don't live long in our populations. Whereas the tiny little cuts in the right combination are clearly easy to sneak through generation after generation to manifest into susceptibilities to heart disease, to hypertension, to all of these different things. And so, you know, genes that, you know, when I was a postdoc in my day, NKX 2.5 or Tin Man in Drosophila melanogaster was the holy grail of awesome, because when you knock out Tin Man in a fly, the mice literally have no heart, which is why the cool fly people called it Tin Man. And, you know, we all thought that there'd be like these amazing genes that we would be able to find and discover in the old-fashioned way by doing lambda GT 10 like library screens. Joe's laughing. All the point, you know, so Jeff has had a little bit of hand on this too, where you pour like thousands of agar dishes and grow beige and like, you know, it would
right of passage.
Yeah, I thought you guys had. What are you talking about?
And, you know, when we would find these genes and we'd knock them out and the phenotypes would not be these like, oh my God, the mouse doesn't have a head, or, you know, we would be like, oh man, this stinks. But, you know, if you drill down into them, you start seeing these things that are, wow, this is really profound. And, you know, you can, when you're receptive to seeing these subtle changes and these subtle differences, it gives you clues. And, you know, what brought me to my path was more reading the literature and finding these things that other people were doing to give me ideas on what I was missing in my data. So I know that's a roundabout answer, but, you know, to really kind of nail down whether hypertension is a component of this, we would really have to just look at the whole renin-angiotensin system. One of the things I can tell you is both HAN1 and HAN2 are expressed extremely high in the adrenal glands. They play an enormous role in the sympathetic chain, catecholaminergic neurons, not the dopaminergic. So they're definitely make epinephrine. You know, the HAN2 knockout in Neurocrest is lethal at 12.5, unless you give them phenylephrine or isoproterenol as an agonist, a beta agonist, because that allows the heart rate and everything to stay high enough where they can make it. But, you know, so they're playing roles in a lot of organ systems. They're expressed really interestingly in different combinations and borders of tissues. So endocardium versus myocardium, there are different components of HAN factors in those two tissues which make direct contact. Within epicardium and myocardium, there's a unique expression profile of HAN factors. You know, within the placenta, you know, the decidua expresses HAN2, all of the trophoblasts and labyrinth cells express HAN1. And it goes on in these various issues. And somehow they are modulating what those cells need to be and why those cells need to stay there or move is what we're really thinking right now.
Thank you.
So I have more questions, but I want to make sure other people ask.
You're fine. Go ahead.
I don't want to monopolize, but Tony, it was a really great talk. I really enjoyed it. And I see Beth back there. Hi, Beth. I hope she's working and not shopping.
Yes, she can't hear you because I have my headphones in, but
Okay.
Joe Miano is saying hello to you.
Okay, so the SNP data, I mean, that's beautiful. Well, first of all, great use of CRISPR with knocking in flag. I mean, I'm with you with that. With myocardium. I mean, as you know, with myocardium, we've had the same problem, but now we can see myocardium, only we use HA because flag in our hands, I think I told you, didn't work on sections, but HA does. So kudos to you guys for getting that working with flag. The SNP data, I'm most fascinated with that because that's a beautiful translational work. And I'm really curious, you showed the homozygous SNP mouse. Presumably the heterozygous is probably unremarkable or no different than the wild type? Or is there a
Yeah,
subtle?
because these experiments are very time consuming, and because we have to rely on the expertise of our EP people who have their own work to do, we're very fiduciary about the requests we make out to get that data collected. Now we have some funding. So what we're doing, we're finishing up some studies where, because we can detect hand 1 in the adult conduction system, why is it there? Is it playing a homeostatic role? And so we use the contactin 2 Cre to knock out hand 1 at P2.
Okay.
So then all of the congenital problems we have are then not there. And can we see problems in cardiac conduction in adults because hand 1 is not in the adult system? And so we have those optical mapping analysis. We've done the ECGs, and the ECGs are really subtle and not anything that you could put a finger on, but the optical mapping may tell a different story. And hand 2, is hand 2 there? We don't know. We don't have a great reporter line that would tell us that. But what we do have is the ability to knock it out specifically in the left ventricle with our hand 1 Cre. And we know that those mice live. And so what we're going to do is we're in the process, we're doing an embryonic Cre of hand 2, and then we're going to do conduction analysis of that mouse, as well as we are going to knock out both hand 1 and hand 2 in the adult and see if that does anything worse than the hand 1 by itself, and basically just use the gene dosage tricks to see whether we could tease out what it's doing in the adult, because that could be a really important thing for people who have like ventricular conduction abnormalities. Joe is frozen. I wonder if like
frozen.
They turned off his power or something.
He might come back. Okay, so we have about 13 people left in the room. Anybody else have any questions?
Thank you guys for sitting through it.
Thank you for giving it. It was amazing. And let me just reflect on 30 years of progress. My goodness, the way you're using tools these days, it's amazing.
Yeah, I mean, if it wasn't for limited time and money, we would be doing lots of stuff. But, I think for any of the junior people in the room, I always think it's a privilege to be able to sit at a lab bench and do stuff and, talk to people and interact and, work in the lab environment. It can be very, very satisfying when you very slowly, year after year, come up with more puzzle pieces to put in the picture to give yourself an understanding of how this interesting biology works. And it doesn't even matter what biology you're interested in. The idea of being able to discover stuff. You remember when you were doing the pick one work, when you do those yeast 2 hybrids, and you'd be the first person to see that this protein interacted with kinase C. And you're the only person in the world who knows that. And how exciting that can be. It's just been great.
You've worked your way to the cutting edge, my friend. Good job.
Hanging on, kicking and screaming, I guess.
All right. Everybody, have a great rest of your day. Tony, thank you again. Everybody.
Yeah.
Thank you for coming, everybody. Good job, Tony. Good job.
Yeah, that was okay?
Yeah, it was awesome. Inspirational and transformational for many of my colleagues, hopefully. Take care.
Yeah, you too. All the best to the family. Let's try to keep in touch.
And when you drive through Joplin on the way to visit the daughter or Kansas City, take a left turn.
You betcha. All right, man. Have a good weekend.
Take care.